Imagine that you are a patient who recently found out about placental screening, a non-invasive way to screen a fetus for genetic abnormalities. You decide to learn more about two options for screening pregnancies. The first is the traditional method, called amniocentesis, and the second is a newer method called placental screening. In amniocentesis, a sample of the amniotic fluid, or the fluid surrounding where the fetus is growing, is taken, whereas in placental screening, only a blood test is taken. This means that amniocentesis is more invasive than placental screening. The way that this blood test works is that within the mom's blood, there are actually pieces of fetal DNA that are derived from the placenta. When a blood test is taken from the mom's arm, placental DNA can be extracted. This means that it's non-invasive to the fetus. You then decide to look more into how placental screening works. Luckily for you, you come across some resources, including pamphlets discussing the government, industry, society, and science side of placental screening. You start by looking at the science pamphlet. Let's see what we can learn about the science of placental screening. Luckily, we have the help of Giulia Del Gobbo, a PhD student working at the BC Children's Hospital. She works in the Robinson lab. Her project focuses on examining the role of genetic variation in the placenta and its relationship with pregnancy complications. Now let's go chat with her. Giulia, would you mind telling us a little bit about your project and what you're currently working on?
So I'm currently a PhD candidate in the medical genetics program at UBC. So I'm doing my research in Dr. Wendy Robinson's lab, and we study mostly the placenta. We've had some other research projects studying kind of pregnancy, recurrent miscarriage, some studies in fetal tissues and that sort of thing. But mainly we focus on the placenta and trying to understand a bit more about how the placenta works, what goes wrong in terms of like pregnancy complications. So trying to understand genetic variation in the placenta and how that may influence how the placenta functions and therefore risks for different pregnancy complications. So my research specifically is focusing on genetic variation in the placenta and how it's related to fetal growth restriction, which is kind of this poor growth of a baby during pregnancy. And this can pose some health risks both during pregnancy and beyond for the babies if they don't really reach their growth potential.
Now let's look at how Giulia got to working in this project in the first place. She started by doing a Bachelor's of Science at Queen's University in biology. Then she took a number of genetics courses and life science courses throughout her degree. She really wanted to do a PhD in fetal development, which led her to Dr. Robinson's lab, and finally the UBC Medical Genetics program, where she is currently. Placental screening not only looks at the genetic information within your cells, but also environmental epigenetic markers. For example, let's say we have this piece of DNA that contains this blue gene here. This blue gene can then code for an mRNA, which can then be made into protein. Say that normally this mRNA is not made that often. However, let's say then we have an epigenetic modification that comes in and modifies the number of mRNA molecules that are produced. Now we see more mRNA being made, meaning that more protein from that gene will also be made. This allows for researchers to explore DNA within a placenta in more than one way. Now back to Giulia. When and how are these tests conducted?
In our lab, to study genetic variation, we also study a lot of epigenetic variation, so kind of these chemical modifications to the DNA sequence that doesn't actually change the sequence but can change the way that genes are read and expressed. So to study those, we use a variety of different techniques, from very simple, what you learn in undergraduate, like PCR, so polymerase chain reaction, just to amplify specific regions if we're really interested in like one small mutation.
The polymerase chain reaction, which Giulia just mentioned, is used to amplify specific regions of DNA. This way you can take one sample and make it into millions of copies of the same piece of DNA. Another technique is gel electrophoresis, which visually helps you tell the difference between genes by their size. Next up are microarrays, which help identify a variety of gene segments from a sample. Aside from the lab experiments themselves, it's also important to understand the accuracy of these tests. Microarray chips show placental DNA expression. This information can then be sent to a computer program where bioinformatic analysis can occur. This software can help determine the accuracy of the results. Accuracy is explored in two main ways: technical replicates and bioinformatic analysis. To do technical replicates means doing the same experiment multiple times, whereas bioinformatic analysis is comparing results from many samples and replicates to create a baseline. Despite all of these tests, it can still be hard to determine if a test result is accurate or not. Back to Giulia. How are these tests used now, and how do you see these tests evolving in the future?
Right now in the clinical setting, they're doing this mostly for testing for chromosome abnormalities. So trisomies, so what we kind of think of more commonly as like trisomy 21 Down syndrome and some other more common trisomies that happen during pregnancy. But actually this kind of cell-free fetal DNA that we're testing for in mom's blood is actually derived from the placenta, because the placenta is what's really directly interfacing with the maternal blood. So, the placenta is what transfers oxygen and nutrients to the baby from the mother's blood. So what can happen is, some cells can shed from the placenta. And so basically the DNA that we're finding in mom's blood that's, labeled as fetal is actually from the placenta. So really we're screening the placenta for chromosome abnormalities when we're looking to screen for, fetal chromosome abnormalities. And I think this field, currently it's, we're only testing for a few things, but it is starting to be a lot more widely used in the clinical setting.
This leads us to our main question of our science segment. What are the pros and cons of non-invasive placental screening? Next up, let's look at the industry pamphlet. What can we learn about the industry of placental screening? The idea behind placental screening is that there's fetal DNA in the mom's blood that comes from the placenta. Therefore, the mom's blood can be tested. Then, when looking at the samples of the mom's blood, we can test the fetal DNA. This is a drastic change from other types of prenatal tests that require a lot more invasive screening techniques. One of the largest applications of this technology is in sex determination. Placental screening would help determine sex at earlier time points in pregnancy and with more accuracy. These placental screens can help determine if a child has two X chromosomes or an X and a Y chromosome. However, here industry might come into contact with another problem. Who gets to share this information? In the traditional ultrasound sex determination process, it is the physician's role to tell the sex of the baby. If there are private companies offering this blood test and placental screening, who will be responsible for delivering these results? Furthermore, if characteristics such as sex are being determined through placental screening, what other characteristics can we screen for? We may be able to screen for hair color, genetic diseases, and even eye color, among other physical characteristics. What effects will screening for these types of characteristics have on our patients? This leads us to our main question from our industry section. How should industry balance the need for profit with consumer protection and privacy? There are a lot of questions about placental screening, including who can get these tests, when can people use these tests, and how do we regulate which tests are used? One large question is, what should the government's role in placental screening even be? Now let's ask Giulia, how does society affect what research you can do?
Cell-free fetal DNA testing for non-invasive prenatal screening. The government is certainly involved there, and it will probably, if we are going to have more widespread testing, perhaps beyond just testing for a few chromosome abnormalities, then certainly I think there will have to be collaboration with kind of the research side and government and the clinical side of kind of aspect, and just kind of organizing regulations for what else we would potentially be testing for. Because of course, there are a lot of ethical considerations when we're doing any sort of prenatal screening or testing. And so that definitely has to be taken into consideration. And the experts are definitely going to have to be involved in these discussions if we want to start testing for more things prenatally. We're pretty lucky to be doing this kind of research in Canada. We have a relatively open open-mindedness toward collecting placental tissue, particularly from earlier time points in pregnancy. And this is really valuable to us because we can learn a lot about how the placenta develops early on in pregnancy, and a lot of complications that arise, are caused from like issues that happen early on in pregnancy. So it gives us a window into earlier development. So that is, a really nice thing about doing this kind of research in Canada, at least.
Now that we know how much the government is involved in regulating placental screening, what characteristics would a government deem acceptable to screen for? Thinking back to the characteristics we discussed earlier, would they allow screening for genetic disease, eye color, and who would get to decide, the parent or the government? Looking at the government's role in regulating placental screening, we can ask the question, for what types of genetic characteristics is it appropriate to use placental screening? Now let's move on to our final section, exploring how society interacts with placental screening. The way that society perceives different topics on research, screening for fetal trisomies, and sex determination can all change how these placental screens are done. Does society think it's acceptable to screen for sex, genetic disease, and physical characteristics? What about intelligence and athletic performance? How do these characteristics change what patients do differently? And do some of these characteristics require further testing in order to understand? If you'd like to learn more about using genomics to predict athletic performance, check out this podcast, Understanding Your Game, Sports, and Genomics. Another important idea to discuss is that if we did screen for these characteristics, would they even be actionable? What impacts would they have? Furthermore, how can we ensure that this information is used in an ethical manner? What is the future for these patients after the placental screening has occurred? As this test can diagnose genetic abnormalities within the fetus, this test can help pregnant women, as well as women who have struggled with their pregnancies. But who will have access to this technology? Will it be something that each individual has to pay for themselves? If so, this means that not everyone will have access to this technology. If this is the case, should anyone have access to it? This leads us to our final question. What consequences might there be in society if placental screening is not equitably implemented? Today we've covered a number of topics related to placental screening. We've touched on the ways that placental screening interacts with government, society, industry, and science. Now I want you to think, what do you think about placental screening? Thank you for watching this Gene School video. If you'd like to see more Genome BC educational content, go to genomebc.ca/education. Thanks for watching!